Okay, I'll go ahead and turn on the recording and share screen. So we left off last time talking about viruses, and we had described sort of the lytic cycle, right, which is quite common in the environment, right? So a good strategy to survive for microbes is to have a, if you have a good source of hosts, then getting in, replicating yourselves, and killing your host, and spawning a huge number of virion particles from that host is a great way to do it. But if we think about it in the environment, oftentimes things are not so great, right? We've talked about how energetic needs, metabolic needs are often exceeding what's available in resources in the environment. And so viruses need hosts, and fairly plentiful hosts, in order to survive, right? So they're tied so intimately with their hosts. And so they've evolved ways of adapting to this. And so an alternative life cycle, which is really, it's kind of adding on to the lytic cycle, because as you can see on the left-hand side here, these viruses are capable of a lytic cycle, but they're also capable of becoming so-called temperate phage or lysogenic phage. So this reflects the fact that they don't kill their host right away. And so what can happen is when the virus injects its DNA, it can either immediately go through the lytic cycle, but if conditions are not very good for growth of the host, for example, there will be certain cues which will cause the temperate phage or the lysogen to become what's called a prophage, right? And they insert themselves into their host genome. And then they're basically just like on a bus getting a free ride, right? So they're in the genome, the host will replicate, form daughter cells, and each of those daughter cells should get the lysogen DNA in it as well, right? So as you can see, this is often for DNA viruses, right? Because it makes it easy for them to go ahead and get in and integrate their own DNA into their host's genome. Now, over time, they can just sort of replicate and replicate. And oftentimes what happens is that the virus will sense if the host is not doing very well, like maybe it has ceased growing, or it has sustained UV damage, or certain things like this, which could impact the virus negatively, and the host, of course. And then they will pop out and induce a lytic cycle. And if this happens collectively, then all of those daughter cells that have the prophage inside them will essentially become lytic viruses and burst out. Okay. So that's the alternative strategy. Um, and it's likely that this more complicated system evolved, out in the environment in these kinds of, where conditions vary and fluctuate so much. Now, anytime we have an evolutionary cycle like this in terms of viruses evolving to infect hosts, just like with antibiotic resistance that we talked about earlier, viral resistance definitely evolves as well. And so there are a number of strategies or a number of ways that, like bacteria, that are mostly those that have been studied in this regard, have evolved to defend themselves against viral infection. So and these can be roughly divided into pre-infection and post-infection strategies. So before infection, you can stop the virus from adsorbing to the surface, and this can involve things like production of EPS, those extracellular polymeric substances, which stop viruses from even contacting the cell. Or you can have a system like the superinfection exclusion system, which stops them from injecting their DNA into the host cell. Another strategy can be that if the viruses have already entered your cell, that you can then remove them or inactivate them. And so one way to do that is through restriction enzymes, right? So you guys have learned about restriction enzymes in genetics class or in cell bio class, right? What does a restriction enzyme do? Who remembers?
It cuts DNA at certain portions of it.
Exactly. And so these restriction enzymes likely evolved to keep out foreign DNA. And of course, viruses have foreign nucleic acids. So these enzymes will recognize certain motifs, and we now use them for genetic manipulations and biotechnology, but they evolved in this context. And then there's the CRISPR-Cas system, which is another example of something we now exploit. And it's arguably the most important genetic engineering tool that has been developed in the last decade. Nobel Prizes are being won for this. And however, it evolved as a defense against viral infection. And these are these repetitive genetic elements that also act to stop insertion of like a prophage into the virus or the host's genome. So a number of ways that resistance has evolved. And you can actually see this in cultures. When we look at cultures, sometimes they will suddenly no longer show evidence of being infected. And that's because some mutation occurred which enabled them to evolve resistance. And that's often going to be at like the recognition, right? We said there's a very important recognition between host and virus, and those mutations accumulate pretty rapidly as well. So understanding the diversity of viruses is even more complicated than understanding the diversity of hosts. And we said that's considerable. Remember last time I mentioned there's no 16S gene for viruses. And, we have both RNA and DNA, single-stranded, double-stranded, as is shown here. And so it's very complicated. Additionally, you can't use morphology to define because there's a lot of different similar things, like we saw those head-tail phages that exist in different classifications of viruses. But thankfully, we now have molecular approaches. And so this is just a fairly recent paper from about 4 years ago, which at that point was a review of the different viral groups. And these are mainly looking at families. You can see there's a lot of families here that have been identified, and using, in this case, amino acid gene sequences, protein sequences, they created this tree, which is very similar to trees you guys will have seen for the hosts, for bacteria and archaea. So this is essentially where we've gone recently with the advent of molecular sequencing. So metagenomics, which is sometimes referred to as metaviromics in this case, can be employed. So you can do so-called cultured viruses if you have them in culture with a host, or you can go out in the environment and study them directly from the environment. All right. So because I care about extremophiles, I always like to share this, and they're really cool. There's some really interesting things like these bottle-shaped viruses. A lot of the extremophage or extremovirus work has been done in hot springs. And so you can see there's these lemon-shaped guys, these filamentous things. But we've done some work in my own lab, so I always like to share this. So I have a former master's student, Megan Rodella, who's a lecturer in our department. Some of you may have actually had her as a TA for general bio. And so Megan, for her master's thesis, characterized a head-tail virus that infected a halophilic bacterium in the genus Salicola. And so these are some TEM images. You can see the viruses all over this host rod. So pretty much as we've said, there's a virus that infects everything. And so if you go looking hard enough, you can find them. And that brings me to kind of the sense of the ecology of viruses. So generally, there's going to be a virus or more for each host. And actually, there's total number of viruses are probably 15-fold higher in viruses, right? So in theory, this represents the largest abundance of organisms and biomass on Earth. And the host range varies, and this reflects the fact that some viruses will infect more than one host, right? And then hosts will often have more than one virus. Just like we have a bunch of viruses that infect us, the same is going to be true of bacteria and archaea and microbial eukaryotes. So this contributes to this, multi-factor, order of magnitude higher numbers of viruses. We've mainly studied these in aquatic and marine environments. We know a lot less about soil or interesting environments like sand and sea ice, but they're undoubtedly there. And as we already talked about, remember, with the microbial loop, viruses are one of the two main causes of mortality among like the bacteria plankton that plays such an important role in the ocean, right? What was the other source of major mortality in addition to viruses? Anybody remember?
Protozoa.
Yeah, so those little nanoflagellate protists were the other major ones. So, and an important thing to think about is their role in lateral gene transfer. We've mentioned that the process whereby viruses transmit genes from one host to another has a name. Does anybody remember what that name is? It's not conjugation and it's not transformation. It's transduction. Yeah, thank you, Angel. So transduction plays an important role, and it's likely to be really common among the lysogenic viruses because remember, they're inserting themselves into their host DNA, into their genome, and then popping back out when they undergo lysis. When they go in and they pop back out, sometimes mistakes are made. And that's where grabbing genes from a host, popping out, and then when they insert in a new host, that can lead to lateral gene transfer pretty commonly. So one of the other really cool things to think about with viruses is something called phage conversion, right? And this is typically, again, for lysogens. So phage conversion describes the situation where the virus contains genes that the host doesn't have. But when the virus is infecting and is inserted into the genome of the host, those genes can be expressed and confer new phenotypes on their host, right? So an example, which actually we should care very deeply about, is in the pathogen cholera. So as those of you that are micromajors, I hope you're aware, if you've taken pathogenesis at least, cholera, the etiological agent of it is Vibrio, right? Which is a bacterium, right? And it turns out that Vibrio cholera are very common in aquatic habitats. They actually seem to be transmitted by small crustaceans like copepods, very commonly in estuaries. But it turns out that the Vibrio cholera is not the pathogen that we normally think of, this lethal thing that causes, among the highest mortality due to pathogens on the planet. It turns out that the pathogenic aspects of Vibrio cholera are due to phage conversion and genes that are carried on the CTX phage virus, which infects the Vibrio cholera and makes them a pathogen. So that's a fairly negative example, but there's also examples of viruses, for example, bringing photosynthesis genes into cyanobacteria and changing their photosynthetic profile and their pigmentation and things like that. So it doesn't always have to be involving pathogens. All right, so the last topic on viruses I wanted to cover today is about how we isolate them. If you go to an environment and have a sample, let's say we go to, like the example of my master's student, she went to a hypersaline saltern and she grabbed some water, she brought it back to the lab. And if you want to know how to isolate viruses, one of the best things to do is to grow the potential hosts from a water sample, in this case. So she isolated some bacteria and some archaea from the saltern that she was studying. She then took the water sample, went back to the original water sample, filtered it down, and used that as a potential source of virus particles, right? The virion particles can last for a long time in those. So those water samples were stored in the fridge. She came back to them later and essentially went about trying to isolate a virus that could then infect the host that she had already isolated. And so the way to do this is to use filtration, right? So if you filter using like a 0.2 micron filter, that should trap all bacteria. And incidentally, it'll trap really large viruses, but small viruses, those nanometer sized viruses, should filter through. Okay, so the viruses will go through, but the bacteria and the archaea and the microbial eukaryotes will be trapped on the filter, right? Because the average size of a bacterium we can roughly say is about 1 micron in diameter. So using a 0.2 micron diameter filter or pore size will be good for that. So this becomes what you can call filtrate, okay? And this filtrate then can be used as a source of virus. So we can then perform what's called a plaque assay. And essentially what we're trying to do is kind of the opposite of when you look for colonies on a plate when you're trying to culture, say, bacteria. In this case, what we do is we grow lawns of a host, and you're looking for clear spots, which would indicate lysis of the host. So what we're really going to go after here with these plaque assays is predominantly going to be lytic viruses. And so what you can do with a plaque assay is you can count and you can isolate these lytic viruses. And so just like with a colony forming unit that we described for bacteria, right? We don't say that they're for sure just one cell because there might be two colonies side by side that grow up. We also use the term plaque forming unit because it could be that there's two viral particles right next to each other that cause a single plaque to be formed. So what you do is you use what's called top agar and a Petri plate. You've got your phage solution, which was the filtrate that I showed you 2 slides ago. And then you've got your bacterial culture or your archaeal culture if you're interested in looking at archaeal viruses. And we're going to mix all of these things together. So what you end up with is mixing the top agar, the bacterial culture, and the virus, and you shake it all around, and then you pour it on top of a thin Petri plate that already has a little bit of an agar media on it. And then this so-called top agar goes over the top of that. And the top agar is what we're going to then look for clear spots, and those are the phage plaques. And so this is a picture of one of that master student, Megan's, from her project. And so you can see, it's hard to see the lawn, but essentially there's bacteria all over the surface of this. But where these little clear spots are, that's where the viruses were. And so you can use this, as I said earlier, to count these clear spots, and that gives you an estimate for how many viruses you have in your sample. And you may want to use dilution series and things like that. But the other thing you can do is grab the plaque, extract it, and then that should be full of your virus of interest. Okay. So that is how we isolate viruses from the environment. Any questions about that? All right, so that's our microbe-microbe interaction examples. And now we're gonna, over the next couple of lectures, we're gonna talk about microbes and so-called higher eukaryotes. So the first thing we're gonna look at today are things that are, of course, near and dear to our interest, and we're gonna look at animals. Okay. And then we'll be reading our paper for Wednesday. Hopefully you already had a chance to read that, but please get your summaries uploaded before class on Wednesday by 10 a.m. And what you're going to do in that paper is learn about those bees and their genomes. And today we're going to learn about some other examples. Okay. So just as we talked about with the microbe interactions, we can classify these as being positive interactions, negative interactions, or commensal interactions. Negative interactions, or so-called antagonistic interactions, go both ways. Um, so for example, predation on microbes, we just talked about that a few minutes ago, right? If the nanoflagellates lyse a bacterial cell and slurp up all those, goodies from it, that's going to be predation. But one of the issues there is that you either have to be really tiny, like a nanoflagellate, infecting specific bacterial cells. But if you're larger than that, then feeding on bacteria can be really a challenge because they're so small. And so another strategy is to concentrate them. So one way to do that is to go after where they're already concentrated, and that would be a biofilm. So if you graze or eat biofilm, you're going to be getting millions to billions of cells of bacteria and maybe algae as well. Okay. Another strategy for this is filter feeding. Many marine invertebrates in particular are filter feeders, and they tend to eat bigger things like algae. However, in some cases, remember we talked about like marine snow and particle attachment? So if you have bacteria that are growing on a particle and then that particle gets ingested by an invertebrate that's filter feeding, then that can be digested and provide nutrition for that filter feeding animal. Another process that is a little bit gross to think about, but is a strategy that's really important for some small mammals, particularly things like rabbits, and that's called coprophagy. You're essentially re-ingesting your own dung, right? So they'll eat food, and oftentimes this is important for herbivores, right? Because they'll eat plant material, digest the plant material, but plant material is hard to digest. And so they rely on microbes in their guts to help break down the cellulose and things like that. But the other thing that they can do to enhance their nutrition is to let their dung sit for a while and then go back to revisit it essentially. And then they can consume that. And there was this interesting study published last year. And what they found in that study was that coprophagy helped to stabilize the gut microbiome of these types of rodents and rabbits and things like this. It helped promote microbial metabolism because remember what happens is the dung is filled with microbes, right? Just like all fecal matter is. And after the dung is out in the environment, those feces, those bacteria will grow and divide. So when you re-eat it, it then can provide more energy for you and more microbial biomass to consume. And that study even found that it helps cognitive performance in these rodents. So they studied those that were allowed to eat their dung and those that weren't, and they found that they were smarter. So I thought that was a really fascinating study. Now, of course, there are also, it goes the other way, right? As we well know, disease and predation or, you know, death of animals based on microbes is definitely something. And so parasites are a big deal. Toxin production, right? So we've talked a little bit about red tides before, right? So the photosynthetic algae that, especially when they're in blooms, lots of algae producing these toxins can lead to problems for their filter feeders, but oftentimes it's the animals that eat the filter feeders that end up really being poisoned because the filter feeders concentrate those toxins in those red tides. So that's why we have to close shell fisheries when there's been a red tide because there's a high danger of the neurotoxins and other toxins that are found in those red tide algae being concentrated by things like mussels, oysters, etc. And of course, we and many, mammals, all mammals essentially, have evolved complicated immune systems. And of course, invertebrates have a simpler immune system as well. The reason that evolved, of course, was because of all these pathogens that infect us. Okay, and so that the entire field of immunology essentially reflects the fact that there's this interplay between host and parasites. So I'm not going to spend much more time on that because we mainly are interested in non-pathogenic organisms here. So let's talk about commensalisms, or most of these examples are actually going to be what we call mutualisms. It can be hard to find true commensal relationships where only one organism benefits and the other one doesn't. So one example is under the category of microbial gardening. Then there's also a couple of other examples, digestive commensalisms, right? Or mutualisms. We're going to talk about the cow rumen. We're going to talk a little bit about termites, although I don't really have as much time, so I've kind of cut some of that out. But termites also have mutualistic associations with microbes. And then I will touch very briefly at the end of the lecture. We may not get to it today, but we'll talk about that with us. Okay. There's also endosymbiotic interactions where there's actually intracellular parasites, or not parasites, but mutualists that live inside their host, in the case of the insect aphid, as well as things like the squid that have a non-pathogenic version of Vibrio that are luminescent with them. So let's talk about fungal gardening. So it turns out that insects often will essentially farm or garden fungi. And so what they'll do is they'll inoculate vegetation. So these are typically herbivorous, so-called herbivorous insects like leafcutter ants that go and collect large amounts of vegetation. They will then inoculate the vegetation with a fungus that they keep in a very special organ called a mycotangia, right? And remember, every time you see MYC, that refers to like mycology, to the field of fungi. Okay. And then what happens is the fungi will be sort of morphologically altered. Um, and then they're essentially almost like you know, beholden to their host. They get inoculated to it. And then the insects can then go back and eat the fungi, right, as a nutritional source, in addition to eating the vegetable material. The other thing is that the fungi are decomposers, right? So they break down, they have cellulase enzymes and these key enzymes for metabolizing leaf material. So the eating the fungi vegetation mixture gives you the biomass from the fungi, as well as growth factors and the plant material that's been broken down. And then interestingly enough, if they eat these fungi, they can store the cellulase enzymes and those other degradative enzymes in their guts. And so they can then go out and eat vegetation without the fungi associated with it for a period of time while they still have those enzymes in there, and use that for their nutrition. Eventually that runs out, and so they'll need to go back and refeed on their fungal biomass. But it's a really elaborate system. And so this is an example. Uh, in the tropics, there's many species of these so-called attine ants or leafcutter ants that have elaborate nests where they bring back vegetation. And you can see all this white, and you can see it here too, right? This is just dominated by fungi. And so these are all in a single genus called Atta. That's why they're called attine ants. And it's thought that this evolved at least 50 or 60 million years ago. There's over 200 species of ants in 13 different genera, and all of them have these fungi that they associate with. Okay. And often in many of these associations I'm going to talk about today, these are very specific relationships where you have a host, insect in this case, and its fungal symbiont that live together and have co-evolved together over these millions or tens of millions of years. There's other insects that do this as well, things like ambrosia beetles, and I mentioned earlier termites. Now, termites, as you know, eat wood material, right? Of course, we hate them if we get them on our house. But one of the reasons they're able to do this is that they have fungal and bacterial symbionts and even archaeal symbionts that help them to digest the material. So this is just a picture of, this is one of those beetles that I was mentioning. So it's not an ant, but it's a beetle. So this has evolved multiple times in different groups of insects, because not all insects do this, and not all members of the attine ants seem to be a radiation, probably from a single species that has radiated and evolved and diversified over time, most of which are going to have these associations. But some of these other beetles are related to beetles that don't do anything with this fungal gardening. But some of them have, and you can see that they've evolved these organs. So here's that organ again called the mesotangium. And you can see it's not far from their mouth parts, right? And so essentially what happens is they can use their mouth parts and grab a little bit of the fungi and inoculate. In this case, this is a beetle that bores its way into trees. And so a sort of sad example of this is chestnut trees, right? So there used to be real chestnuts all over the United States and Europe. Um, and what happened was these beetles started spreading a fungus called chestnut blight. And these beetles have essentially spread all across North America, infecting most of the chestnut trees. And so these days, the only chestnuts you'll find in most locations are called horse chestnuts, and they're not even real chestnuts, right? So that old Christmas carol, right? Chestnuts roasting on a it's really hard to find chestnuts now because of this pathogen. So you can see here, this is an example where I think these are probably larvae of the beetles that are living in this hole that was created by the adult. So the adult inoculates the tree, the fungi go crazy growing here. They then lay their eggs, they mature, and they have food to eat in terms of the plant, the host tree. All right, so that's one example or one set of examples. Any questions about fungal gardening? All right, so the next topic is the rumen, right? So the rumen is another example of one of these elaborately co-evolved systems, right? So most mammals have guts kind of more like ours, right? Essentially a single tube that runs from the mouth to the anus. And there may be intestines, there may be a stomach, right, et cetera. But usually it's like a single chambered stomach, et cetera. But herbivorous mammals, things like cows and goats and deer, right, they're herbivores, right? They have a high cellulose diet, they eat plant material. And in those cases, right, they face the same problems I was just talking about with the insects, right? So mammals don't really make cellulase enzymes. And so if you're going to eat a high plant diet, you've got to break that down somehow. And so what has happened is they've evolved a more complicated stomach with multiple chambers, right? And so that's shown here. So there's, instead of having just like a single bag where you might have acid like we do, right? Where things go in one side, they get bathed in the acid, and there's, mechanical sort of muscular contractions, things like that. And then it moves on to the intestines. In this case, what happens is you have multiple chambers and specialized almost micro niches for microbes. Now, like our guts, these are going to be anaerobic, warm environments, right? Cows are a little bit warmer than us. We're about 37, they're 39, but same difference. And it's going to be about a pH of 6.5, which is fairly near neutral, right? And we're going to come back in a minute to how that's maintained. And what happens is the cows or the deer or whatever will eat vegetation, swallow it into the stomach, where it then gets sort of mixed around in the rumen, right, with lots of microbes. There's going to be essentially microbes from all three domains in there that are working on it. And then what they do is, you've probably heard this expression, chewing the cud. So what that is, or it's also called rumination, because it kind of looks like they're chewing themselves, but they get this serious look on their faces and they sort of look like they're thinking. And that's where the term that is sometimes used for us is called rumination. But essentially what they're doing is they're regurgitating this into their mouth and grinding the cud, which is essentially a mixture now of the vegetation matter and all the microbes that are found in the uh, rumen. And then they're mixing it with saliva from their mouth and essentially helping to physically and mechanically break down the vegetation in concert with the microbes that are living there. So when you look in a rumen, and because this is agriculturally relevant, a lot of schools, particularly agricultural schools, so oftentimes, Davis is the UC campus on in our state that's the major agricultural school, but in many cases, states will have one major school and another one. And oftentimes it's the one that's like such and such state, right? That often started as an agricultural school or a land grant school. And so in those schools, there's often lots of agricultural research that goes on. And that's certainly true at UC Davis as well. So in these places, and of course, in places like the Midwest where agriculture plays such a huge role, like the University of Iowa and Iowa State both probably have massive amounts of agricultural research that go on. So you can imagine the rumen is of intense interest to farmers, and so it's also of interest to researchers at those schools. And so rumens actually have been studied very well. Um, And I won't really go into the details of how you study a rumen because you can imagine you got to go in either one end or the other. Um, but I've even seen pictures of some animals that have been modified so that they have kind of ports so that the rumen can be sampled on a fairly regular basis. And through that research, what they've found is that there's an incredibly dense and abundant microbial community, upwards of 10 to the 11th cells per mil, right? The organisms that live there depend on the diet, and the diet is really key. So farmers will feed their cows very specified diets, right? And if you change the diet, it can actually have a profound impact on the microbes that live in the rumens. So they don't do that lightly. Okay. So among bacteria that are there, there's lots of genera like Ruminococcus that you can imagine. The first organisms isolated there were isolated from a rumen or Ruminobacter. Many of these are going to be fermenters because, of course, I mentioned earlier that these are anaerobic environments, right? So just like in our own guts, we have lots of fermentative organisms. The rumen has lots of them, but many of them are specialized, right? So the reason you find something called a ruminobacter there and you don't see it in our gut is that it has evolved to be in this specialized organ. Among the archaea, by far the most important organisms are methanogens. And so methane production is a huge issue with regards to livestock cultivation. And I hope many of you are aware that methane, just like CO2, is a greenhouse gas. And so it's a really big issue because it turns out that cow belches and cow farts are a substantial source of methane in the atmosphere and growing, right? And one of the reasons it's growing is that as developing nations like China switch from a vegetarian based diet to a meat based diet, they are now, cultivating many more cows and other ruminant organisms to be eaten. right? And if all of these ruminants are producing methane, the more cows there are on this planet, the more methane that's going to be produced. So this is a really big issue and an active area of research in terms of trying to figure out ways to maybe shut down the methane production in agricultural livestock. In addition to the two prokaryotic groups, eukaryotes are also important. We've got protists, especially things like ciliates, right? We've got, and there's, as I already mentioned, there are protists that have methanogens or have bacterial symbionts that live inside them, inside the rumen, inside the cow. So it's really an interesting ecosystem. And many of these protists are going to be preying upon or eating and consuming the many bacteria and archaea that live in the rumen. There's also fungi, which contribute those cellulolytic enzymes, just like with the insects. And so there's often this very complicated relationship. So why did this evolve and what's going on here? Well, essentially, it's all about degrading the the vegetation that these animals eat into usable um, carbon sources um, and food, right? And it turns out that many of the products of this um, are able to uh, be transmitted to the host. And many of them are in very small packages as a volatile fatty acid. So those of you taking um, bacterial physiology this semester, have you guys talked about fermentation products yet? Yes, no, maybe. So fermentations are often named based on the product that comes from it, right? So we have terms like acetogenic fermenters, right? Or um, that is, there are specialized groups of fermenters that will collectively produce many different kinds of these short chain fatty acids. Okay. And these are referred to as volatile fatty acids, and they're very labile. They can move around. Okay. And so this is just a diet or a stoichiometric equation that's basically showing the glucose, right? Which of course cellulose is just a big string of glucoses. For every glucose, you can generate, or every almost 60 glucoses, you can generate a bunch of acetate, propionate, butyrate, all these really lovely smelling things if you remember your organic chemistry labs. There's also lots of gas, right? So there's both CO2 and methane produced. Methane by the methanogens and the CO2 by the bacteria primarily. And so then they have to get rid of the gas, and so they often will belch it or eructate that out. Now I see a question from Robin in the chat. Are the protists acting in an immunological capacity? I would not say that's the case. I think essentially it's like an ecosystem inside. They are, they may be killing some of the bacteria, but they're not doing so necessarily in a selective way, right? Because they're phagocytic, right? So they will roam around killing anything they can get their hand on. And of course, these are symbioses. So the bacteria and the archaea that are living in there are actually benefiting their host. So it's not quite what I would call immunological. So what happens is all these volatile fatty acids or VFAs get produced, okay? And they often then cross into the bloodstream. So they'll cross the barrier of the rumen, the organ, tissue walls, and they'll move into the bloodstream where they can be oxidized. Okay. And then, of course, other elements from the rumen move into the other chambers, right? So I didn't go over all the titles and you don't have to know, but there's the abomasum, which is essentially the stomach. Um, and, there's further digestion that goes on just like it does in, other animals. And so these are going to provide carbon, amino acids, vitamins, etc. to the host. Now, the crossing of the VFAs across into the bloodstream serves two purposes. One is those volatile fatty acids can be used for nutrition. They can be oxidized, and it's, good carbon. Okay. But I have a poll question for you. There are some other possible benefits of having this crossing into the bloodstream of these VFAs as well. So see if you can figure out what I'm coming at. So let's have our first poll. So which of the following would be a benefit of the transfer of these volatile fatty acids across the rumen wall into the bloodstream? Would it lower the pH of the rumen? Would it provide nutrition for the cow? Would it stop the cow from inhaling bad vapors? Would it provide vitamins to the cow? Or E, all of the above? So let's go ahead and launch the poll. All right, is that everybody? Go ahead and log your answer if you haven't already. All right, so let me share the results. All right, so a bunch of you thought they all sounded really good. Quite a few said B, which of course is what I had said too, but so let's dive into this a little bit. So B is absolutely correct. Okay, it provides nutrition for the cow. So let's look at A, C, and D to see if E was actually correct as well. So volatile fatty acids are leaving the rumen and going in the bloodstream where they get oxidized. So what effect do you think that would have on pH of the rumen? And essentially here I'm alluding back to the fact that, what did we say the pH of the rumen generally was? Does anybody remember?
Near neutral.
Yeah, it was near neutral, about 6.5, something like that. Okay. So A was it lowers the pH of the rumen. Do you think that moving volatile fatty acids out of the rumen would lower pH or raise pH? So if we're removing them from the rumen, what would happen to the pH of the rumen itself, do you think? Does it go up or down?
Go up.
Exactly, right? So if we didn't remove the VFAs, the rumen would become acidic, much like our own stomachs are, right? But by moving them out of there, essentially they remove this problem and they keep the pH at about 6.5. This is really important because, as I hope you're aware from your own stomach, we can't support a very diverse microbial community at lower pHs. Most microbes are not acidophiles, for example. So it actually helps maintain the balance. And so really the best the correct answer here, if you're talking about pH, is it raises the pH of the rumen. So A is incorrect, B is correct. And so I think you see where I'm going with this. It's nobody put C. That was just a trick question. So we'll just move on. We said that vitamins were provided to the cow, but that didn't come from this piece. The vitamins came from the breakdown of, the fermentation products and things that are produced. Those fermentative bacteria often will produce vitamins and other essential nutrients, but that doesn't have anything to do with the VFAs. Those are not vitamins. Okay, so the best answer was B. Okay. But I just like to have that pulse so that we can talk about this issue, and it's a little more complicated. Okay, but I hadn't told you about this, so don't feel, if it was a little confusing, don't worry about it. Hopefully now you're not confused though. Does everybody see how movement of fatty acids out of the rumen into the bloodstream would raise the pH now. All right, so that's the rumen. But there's so many of these, I can't even share most of them with you guys, but I like to try and hit some of the high notes before we move on to people. So there's also these really cool tube worms at the deep sea hydrothermal vents. You've probably seen pictures of these or watched a special on the Discovery Channel or something, or YouTube or whatever. So Vestimentiferan, that's just a taxonomic term used for it, but these are tube worms, right? And Riftia, so named because it's near the Mid-Atlantic Ridge, that's where it was discovered. Deep sea submersibles like Alvin, which is out of the Woods Hole Oceanographic Institute near Boston, went to visit these hydrothermal vent systems, and they discovered not just the vents and all the cool microbes, but animals living there. And it turns out that the many of the animals there have modified their physiology and their organs to essentially become symbiotic with microbes. So these tube worms don't have real guts anymore. They do have a bag that they probably get, maybe 25% of their nutrition from, but mostly in that gut is found these sulfur oxidizing bacteria. And these bacteria are intracellular. So these are endosymbionts inside the walls of the gut tissue. And so they've actually evolved the ability to bring hydrogen sulfide from the hydrothermal vent and oxygen from the seawater and mingle them together in this organ, because sulfur oxidizing bacteria require both oxygen and sulfide for their metabolism. It turns out there's also mussels or snails or clams that have similar symbioses with sulfur oxidizing bacteria. In those cases, they live in their gills because many of these are filter feeding organisms. So they'll just wash sulfur and oxygenated water over their gills, exposing the sulfur oxidizers to both gases, and allow these chemosynthetic or chemolithotrophic bacteria to fix carbon. So the symbionts fix carbon, and then where do you think that carbon goes? Any idea? If it's a symbiosis, where do you think the carbon is going to go?
To the host.
Exactly. So they will provide, the host is providing the bacteria with a home, and the bacteria are providing their host with fixed carbon. So they don't need to filter a bunch of, food particles or whatever. They can rely for most of their diet on this. And so this is just a picture that shows it's fascinating. They have modified hemoglobin, which binds both oxygen, like our hemoglobin does, and sulfide. And so they can deliver from the plume, this part up here, this is the hemoglobin, you can see the red color. And they bring all of this down into, in this diagram, it was called the feeding body, but this is the trophosome, which is essentially what this is this gut structure, okay, which is filled with, all along the walls of this gut are filled with these bacteria sites that are sulfur oxidizing. So that's one cool example. Another cool example are bioluminescent bacteria. So there are two main genera, the Photobacterium and the Vibrio, that have evolved to become bioluminescent. So these are both gram-negative modal rods. And in the presence of oxygen, they use luciferase enzyme. Now, this is the same enzyme that like fireflies have, although I know we don't really have fireflies out here, but um, it glows, right? In the presence of oxygen, the luciferase enzyme can catalyze this reaction that releases light. Incidentally, this is, if you remember we talked about quorum sensing and bioluminescence. So this is the same thing we were talking about before. There are free living Vibrio and photobacteria that are bioluminescent on their own, and they just glow in the water column. But what has happened is that over time, symbioses have developed where these bioluminescent bacteria that can be, these are just cultures on a plate, put them in the dark and you can see them glow. And this is one of those autoinduction quorum sensing mechanisms. But it turns out that many of these Vibrio and photobacteria have formed associations with animal hosts as well. And so usually these are found in specialized organs inside their host. And you can see from this that there's lots of kinds of hosts and lots of kinds of usage for that. And I will say, we don't know 100% all of the functions, but these are the proposed functions that make a lot of sense. So the first thing that really captures my imagination are these things. So these are one of these angler fish. And where do we find these kinds of fish? Does anybody know?
The deep sea.
Exactly, Daniel. So in the deep sea, right, this has clearly been illuminated by like a submersible, right, or a remote operated vehicle. But generally, this fish is going to be in the pitch black, right? So if you're in the pitch black, if you can have a little bit of light, guess what? That's going to attract some attention by other organisms. And so these anglerfish have this specialized organ and they have bioluminescent bacteria that live at the tip of their organ. And so imagine if you're some unsuspecting little crustacean coming along, checking out this glowing thing, and then like these massive jawed, toothy thing behind it swallows you whole and chews you up. So that's a pretty obvious. I think we're all comfortable saying that is a use of bioluminescence. Where it gets a little, more speculative, but also makes a lot of sense is in other things like mating. So there are some researchers at the University of Hawaii that have studied a small squid called the bobtail squid that only comes out at night and it lives in shallow bays in Hawaii. We should all go there and be bioluminescence researchers, right? So they have studied these bobtail squid for many decades now. And what they have proposed is that these bobtail squid are using the bioluminescence that glows in certain organs. Now, some squid like this one have it all over themselves. The bobtail squid has them in just a couple of select locations. And it's thought that these are involved in mate recognition, right? So you have a male that is flashing these lights and a female will see the lights and be attracted to that and recognize that squid as a potential mate. So this is much like you would see like mating calls in birds and dances and things like that, but obviously you're not going to be dancing and calling under the ocean. So this is another thing that has evolved for these animals. And in this case, they're using microbes to their advantage. The last likely function is involved, it's kind of an anti-predator approach. So this is found in many so-called mid-water fish or shallow fish. And so there you can see there's lots of glowing stuff all over the surfaces of these animals. So this is a big midwater squid. We actually have some of these squid off the coast here. And then this is a fish. And so these glowing things are essentially tiny crypts or bags full of these bioluminescent Vibrio or photobacteria. And what we think is happening is that by causing their undersides to glow like this, it protects them from predators that come from below. So in the midwater, which is, quite deep, but you've got lots of predators that will come up from underneath and they'll actually use the shadow of the animal that they're going to prey upon to tell them where it is, right? It may be dark down there, but if you're looking up, you can kind of see shadows and see things blocking the sun or the moon, depending on how shallow you are. the moonlight or the sunlight. And essentially what the bioluminescence is doing is removing the shadow. And so this is called counter illumination. Basically, you light up your shadow, and so then those animals can't see it. So it's thought that this is an anti-predation kind of response. Any questions about those functions or bioluminescent? Okay. All right. All right. So this one is probably the closest to the honeybee that we'll talk about, but it's quite a bit different. So there's a research group that at one point was at Phoenix University of Arizona and then moved to Yale. Um, and that researcher, Nancy Moran, has pioneered the study of aphids. Now, you might say, why do we care about aphids? Turns out aphids are major agricultural pests. And so if farmers get aphids infecting their crops, it's a really bad news. So understanding how these aphids function was an important priority. But she had no idea when she first started researching this, I'm guessing, that she would discover that aphids are highly symbiotic. But it makes a lot of sense if you think about it. So aphids suck on the phloem or the sap, right? Remember your xylem and your phloem, right? So the liquid, sugary liquid, the photosynthate that the plant is transporting down like the stem or whatever to other parts like to the roots so that they can grow. So it's full of sugars, but it doesn't really have much in the way of proteins or amino acids, right? And so it turns out that aphids, if they pretty much feed exclusively on their plant sap, that's all they eat. And so nutritionally, that's not a very balanced diet, shall we say. And so what seems to have evolved is the aphids, because they cannot get all of their essential amino acids, they don't have 10 essential amino acids if they're on their own. They have evolved an association with a bacterium called Buchnera. And as you can see, it was named for the aphid host, so it's Buchnera aphidicola. So this is another one of these intracellular endosymbionts. So inside their guts, in the walls, the linings of the gut is filled with these Buchnera bacteria sites. So they maybe once were free living bacteria, but they've become these like modified cells. They are no longer free living. They live inside the tissues of their host. And that was true of the bacteria inside the vent worms earlier I talked about as well. So that's why we call them bacteriocytes. Now it turns out Buchnera can take glutamine, which comes from the host, right? And from the sap, they're able to make glutamine. And so the Buchnera take glutamine and convert it into the other 10 amino acids that the aphids can't make themselves. So they give the host, they give the bacteria a home, feed it the sap, and feed it the glutamine, and then they coexist this way. Incidentally, there's even more complicated things here because it turns out that if you look at the microbiome of aphids, they also have another bacterium in there called Hamiltonella. And this is involved in a defense against parasitic wasps. So there are these really cool wasps that lay their eggs inside other insects like aphids. And essentially the wasp larvae end up eating and killing the aphid and take it over, and then the wasps hatch and go on and find a new home. But if there's Hamiltonella bacteria inside the aphid, the Hamiltonella will kill the wasps that are laid inside the aphid. And so it protects the aphid from that. So the interplay between animals and these bacteria is incredibly complicated and fascinating. All right, so the last sort of environmental set of associations are classical ones that you should all be aware of. And these are endosymbiotic algal associations or photosynthetic associations. Sort of the classic one that involves dinoflagellates or so-called zooxanthellae is found in what kind of animals? Do you guys know? Where do we find zooxanthellae dinoflagellates? Anybody know? Really common in the ocean. So these are found in corals, okay? So there's also green algae or chlorophytes that are found in a symbiotic association with animals. These are called zoochlorelli. These are common with some sponges. And then there's cyanobacteria that can be found in some animal hosts called cyanelli. So Cnidaria is the phylum that includes corals, but it also includes, it also includes, right? So this is the class Anthozoa, which has corals, but there's also sea jellies, right? Which we, call jellyfish. That's a misnomer. So marine biologists refer to them as sea jellies. And then sea anemones, right? Those are also in the Anthozoa class, relatives of corals. All of these animals have associations with the zooxanthellae dinoflagellates. Now, these are not free-living dinoflagellates. These are obligate intracellular um, organisms, these algae that they can leave their host, but they have to find a new host to basically survive. All right. Incidentally, if you've ever gone to like the aquarium of the Pacific, if you look at the giant clam exhibit that they have there, they often will shine UV lights on giant clams, and they have their mantle tissue open, and you'll see blue bioluminescence type stuff. Those are zooxanthellae. So giant clams were originally thought to be these like man-eating things, and they even made movies back in the 50s about them. Giant clams are enormous. The reason they're enormous is because of their symbionts. It's not because they eat other stuff. They have these symbiotic zooxanthellae that feed them. So this is just a picture of a coral. And so this is like the animal structure. And as I hope you're aware, there's calcification that goes on. The hard parts of the corals are here. But in a coral polyp, this is the living portion. And these guys have a gut. They actually have, their physiology, basically, they take everything in and out through one hole. And in their tentacles out here, they have little nematocysts, which are those stinging cells which can trap prey. But the other thing that they do, the more important thing that they do, is have these golden reddish brown dinoflagellate zooxanthellae living with them. And these are the photosynthetic organisms. So the zooxanthellae live in the tentacles, in the epidermis, right, the outer tissues. So in this case, the algae fix the carbon, and these corals cannot produce the calcium carbonate reef structures that you guys associate, we all associate with corals, I hope, without that. Turns out that calcium carbonate precipitation is greatly affected by photosynthesis going on near it, because remember, we're taking CO2 out of the water doing this. Now, hopefully you've all seen a picture of one of the really big problems with corals, and that's coral bleaching. So it's thought that coral bleaching occurs when sea temperatures rise. And what this does is, the reason they're bleached is that all of these colored algae die, the zooxanthellae die or are expelled from their host, and it leaves behind the white calcium carbonate skeleton. But essentially, bleached corals are dead corals. They cannot be recolonized effectively, and they will die. And the world's largest reef, the Great Barrier Reef near Australia, has undergone such high levels of warming in the waters there, in the tropical waters where this reef forms. So global warming is raising the sea surface temperature and it's killing and bleaching. So it's thought that the Great Barrier Reef, which is, you can see it from space, it's so big and amazing, and this incredible ecosystem is irreversibly going to die out. So if you want to go to the Great Barrier Reef, go now. Well, after COVID, but go soon in your lifetimes because it will likely die out in our lifetimes, unfortunately. So I already went over the causes. Okay, so temperature. It turns out there's also diseases that can kill corals as well. And these are often fungal diseases or cyanobacterial diseases. Okay. So they can contribute to, unhappy coral hosts and bleaching. But really the biggest culprit seems to be temperature. And it only can take like a half or a 1 degree temperature rise in the ocean. And the delicate evolved balance between the zooxanthellae and their hosts is disrupted. All right, so let's have a poll question. Which of the following would be a example of a nutritional or a food-based symbiosis? Was the aphid buchnera a nutritional symbiosis? Was the cow ruminococcus? Was the squid vibrio, or at least some of the squid vibrio? Or D, both A and B? Or E, all of the above? Which of these would be nutritional symbioses? All right, go ahead and cast your answer if you haven't already. All right, I think most everybody is getting this one. Cool. All right, so most of you put D, which is A and B, and that is the correct answer. So aphid buchnera, what was the nutritional basis for this one? What did the buchnera provide their aphid hosts?
Amino acids.
Exactly. So they converted the glutamine to those other 10 essential amino acids for their hosts. What do the ruminococcus give their cow hosts?
Nutrients.
Okay, so what ruminococcus are probably what kind of bacteria? What are they going to be producing specifically?
They're fermenters.
They are, and so they will produce the what that we talked about.
Carbon dioxide.
Okay, and what else? What's the thing that went into the bloodstream?
Volatile fatty acids.
Exactly. So those volatile fatty acids are common products of, for example, ruminococcus fermentations. Okay, I just want to make sure we're clear on that. Why is squid vibrio not the right answer? What was the squid vibrio implicated in?
Bioluminescence.
Right. And so what was the purpose that the squid were using the bioluminescence for? There were two that I mentioned for squid, neither of which were nutritional.
Mating and the counter luminescence.
Exactly. And I see, Caden, you put mating as well. Thank you. Exactly. Good job. All right. So D was the best answer, both A and B. Cool. All right, so we just have a couple minutes left. I'm going to go ahead and get started on this and we'll finish it up next time. I always run out of time for this one, but I know you guys care about this because it's when we talk about us, and it's the only time I really talk about humans, so I can't bear to cut it out. So I did cut out a little bit. So the skin is also a very important microbiome, and I just don't have time to talk about it this year, so I apologize. So what we'll mainly talk about are the gut and the mouth. Okay, so let's talk about the gut before we close for today. So as I have already alluded to, our guts, like the cow rumen, are filled with microbes, and these are going to be anaerobic microbes. But we do not have a 4-chambered stomach. We do not chew our cud, thankfully, and we are omnivores, right? So we have a very different diet. So everything comes in, it goes into the stomach, And most bacteria don't live at pH 2 like we have in the stomach, right? Incidentally, if you look at all of the, the huge amounts of tubing essentially that we have in our intestines, the surface area is larger than several tennis courts. So essentially you go from the stomach, which is highly acidic, down into the small intestine and then into the large intestine. And what this shows is that there's really a huge increase, particularly as you get into the large intestine, in the diversity of microbes and the abundance of microbes that will live there. Now, this is a very old document that I just show you because it illustrates this, but understand that now that we can do the human gut microbiome, we now know there's much more diversity here than being shown. So just take that with a grain of salt. Up in the stomach, very little can live. As you may be aware, Helicobacter pylori, which is the etiological agent of ulcers, they can live there, but they don't live in the water in the pH 2. They actually live in the lining of the stomach, and they produce urease enzymes, which raise the pH around them by forming ammonia, which is a base, right? So really, there's not much growing in the stomach fluids, right? It's, breaking down that material, etc. And then you get into the small intestines, pH goes up from 2 to about 4 to 5, much more hospitable, still acidic though, right? But we end up with about about 1 million cells per gram on average. But when you get down into the large intestine or the colon, you're going to end up with huge numbers of cells. And of course, we no longer say that this is per milliliter because we're really now talking about per gram of fecal matter. So that's how these things are referred to. Now that we've used molecular sequencing, we know that there's over 400 species that have been identified, and it's probably more now. This may be a little out of date. Many of them are anaerobic fermenters, just like we talked about with the rumen, but they're different species. Many of these are found in a group, the phylum Bacteroidetes, and in this Bacteroides class. But there's also like Enterococcus, which if you're a micro major, you've dealt with before, right? This is just the gut version of Streptococcus, like strep throat, okay? But there's many different groups all throughout the gut, and they all probably play incredibly important roles. And what we've learned over the last decade or so is that everybody has their own individual human microbiome, and they're individualized in some ways, but they have a huge impact on human health, and we don't even know the full extent. But we know that, for example, neurological disorders seem to be connected to gut microbiomes. And so as we learn more and more about this, obesity, of course, is something that's a hot topic in terms of trying to understand are the gut microbes, what role are they playing in these kinds of things? But there's many more human health related things that seem to be affected by these microbes in the guts. So most of these are going to be, well, really all of them are anaerobes. They're either facultative anaerobes or strict anaerobes because of the consumption of oxygen. All right, and I will leave it at that because I've already run over, and we'll talk about the nutritional effects next time. All right. Actually, we'll talk about those after spring break because, of course, on Wednesday we will have our paper discussion. Okay. Any questions about animal symbioses? All right. Well, if there aren't any, I look forward to seeing everybody and having our discussion on Wednesday.